Hello everyone, I'm Dr. Shorban Gupta and we're back with biological classification. Now as we discussed in our previous video, biological classification can be of various types. Now based on a wide variety of criteria, there are three most common systems of biological classification. The two kingdom classification, the five kingdom classification, and the three domain classification. One thing to note is that they're not concurrent, that is, they're not simultaneous, but rather a gradual progression from the two kingdom to the five kingdom and ultimately the three domain classification. This is because as our understanding of living beings has increased, so has the criteria for classifying living organisms. And hence, there have been many attempts over the years from the two kingdom to the five kingdom and even seven kingdom or 10 kingdom. But the most widely accepted ones are the two kingdom, five kingdom, and three domain classifications. Now, first we have the two kingdom classification. The two kingdom classification was proposed by Carolus Linnaeus. It divided all living beings into two groups, plants and animals, and it took into consideration two major criteria, mode of nutrition and mobility. If we consider mobility, plants are essentially sessile. They cannot move from place to place, whereas animals are mobile. They can move from place to place. If we consider the mode of nutrition, then plants are autotrophic. They can synthesize their own food, whereas animals are heterotrophic. That is, they cannot synthesize their own food. Now, plants were further subdivided into two groups, cryptogams and phanerogams, whereas animals were divided into a number of groups. Coming to the cryptogams, as we discussed before, that cryptogams, the word has originated from two Greek words, kryptos meaning hidden and gamos meaning reproduction. That is, these are the non-flowering plants. They were further subdivided by Linnaeus into three groups, Thallophyta, Bryophyta and Pteridophyta. Thallophyta represented a wide variety of living organisms, a huge assemblage of organisms which were all in which the body, that is, if multicellular, was not divided into root, stem and leaf. The body was thallic or thalloid. The Thallophyta were further divided into four groups: bacteria, fungi, algae and lichens. Bacteria, as we all know, are prokaryotes. Now, as we discussed in the previous video, the word prokaryote has come from the Greek words pro meaning primitive and karyon meaning nucleus. That is, these are organisms that lack a membrane-bound nucleus with nuclear pore complex or membrane-bound organelles. Fungi are eukaryotic but achlorophyllous. That is, they do not have chlorophyll, so are heterotrophic, that is because they cannot synthesize their own food. Amongst heterotrophic, these are all holozoic, these are all either saprophytic or parasitic. Now, algae, the third group, refers to a wide array of living organisms that have an equally, if not more, diverse array of photosynthetic pigments and cell wall components and storage, as well as storage products. They can range in size from simple unicells like chlorella to giant macroscopic thalloid structures like the giant kelp or Macrocystis spirifera. Lichens are composite living organisms in that they are composed of a symbiotic association between an alga and a fungus. Coming to the second group of cryptogams, the bryophytes, these are simple or plants which have a rather thallic structure but not fully undifferentiated mass. Here some degree of differentiation is seen, especially in the mosses which have a leaf-like phylloid, stem-like colloid and root-like rhizoids. But these are not true leaf, stem and roots, and they lack vasculature. That is, they do not have xylem and phloem. The pteridophytes, the third group of cryptogams, are also known as vascular cryptogams in that they have true vasculature, xylem and phloem. Here the plant body is fully differentiated into root, stem and leaf. Now, in case of algae and bryophyta and pteridophyta, we get to see a phenomena called the alternation of generations. We'll discuss it in detail in our later videos. Now, phanerogams were divided into two groups, gymnosperms and angiosperms. The word gymnosperm has come from the Greek words gymnos meaning naked and sperma meaning seed. That is, these are naked seeded plants. They include the cycads, the cyclospams, as well as the living fossil Ginkgo biloba and all coniferous trees, coniferous plants like the pines, the cedars, firs or the Christmas tree. Now the angiosperms are flowering plants in that they have flowers. Now the angiosperms are further divided into two subgroups, the dicotyledons and the monocotyledons. The monocotyledons have only one cotyledon, that is primary leaf, whereas the dicotyledons have two cotyledons or primary leaves. In case of monocots, the root system is fibrous, whereas in case of dicots, they have a taproot system. In case of monocots, the floral parts are arranged in multiples of 3, whereas in case of dicots, they are arranged in multiples of 4 or 5. If we see the leaves of monocots, they are narrow. They are narrow and have parallel veins, whereas in case of dicot, they have oval or palmate and reticulate, that is net-like venation. The vascular bundles of monocots are scattered in the cortex, whereas in case of dicots, they are in the form of a ring. The pollen grains of monocots have only one pore or furrow, whereas in case of dicots, they have more than one pore, mostly 3. Thank you, that's all.